Hello, and welcome to the next in our series of lessons on plants for the subject Life Sciences. In our past lessons, we have covered several aspects, and we're going to be building on those aspects with what we are going to do today. We've already looked at plant tissues, plant anatomy, and we started looking at plant functioning. And what we're going to do today is we're going to carry on looking at how plants work. And the topic is we're looking at transport systems in plants, more specifically how water is moved around the plant. The question we're going to be looking at is, when you water a plant, you pour water on the soil. And yet it's the leaves that actually need the water because they need the water for photosynthesis. So let's look at how this works. In our past lessons, we have looked at the structure of a plant, and we also looked at leaves, how leaves lose water through transpiration. The question is, where do the leaves get the water? And that's what we're going to be looking at today, because the water obviously is absorbed by the roots and then it is transported up by the stems. So that's what we're going to have a look at today. We're going to be looking at roots and stems and how they deal with water. To be able to understand this section, you need certain basic concepts from the lessons we've done in the past, namely, what types of tissues do we find in a plant, what do they look like, where they're found. But we also need to revise certain concepts from cells, which is a process called diffusion, and another process called osmosis, which I will revise with you in a short while. Looking at the plant, what we did in one of our previous lessons, we look at roots, stems, and leaves. And in those roots, stems, and leaves, we have the different types of tissues. So let's highlight where these different types of tissues are found. So if we look in the root, we see the dermal tissue. And the most important thing we're looking at now is the vascular tissue in the center, namely the xylem and phloem. If we look at the stem, the vascular tissue is found in the vascular bundles where the xylem and phloem is. And if we look at the leaf, we have the main vein and the side veins, which have the vascular tissue, because it's the vascular tissue that is actually going to be doing the transporting of water. Last lesson, we looked at how leaves lose water in transpiration, and this lesson we're going to look at how does the water get to the leaves, namely through the roots and the stem. To be able to understand this, you need to remember the basic structure of a root, and I'm just briefly going to revise that, focusing on the parts which are important for water transport. So if we have a look, we have the epidermis on the outside with, very importantly, the root hairs to absorb the water. We've got this layer, which is the cortex, and that's mainly for storage. Then in the center, we've got the xylem with the phloem, and then the endodermis and pericycle around the vascular cylinder. In this section, most important, these little things, root hairs. And they're long and thin, and they increase the surface area of the root by 10 times, which means it can actually absorb 10 times more water than if there were no root hairs. If we look at this plant, it is showing a plant absorbing something through its roots. But it's not only water that plants absorb, it's also minerals. And let's have a look at which minerals will the plant actually need to absorb along with the water. So if we have a look at that, okay. From your natural science early on in grade 8 and 9, you should know that K is an element called potassium. Another element in the soil that is absorbed is P, which is phosphorus. And then the third most important one is N, which is nitrogen. And those are the three most important minerals that plants absorb. They do absorb a whole range of other ones, but these are the most important ones. Now, how do these get into the root? Easy. Simple diffusion. And diffusion occurs when substances move from a high concentration to a low concentration. But the problem with diffusion is when concentrations are equal, it stops. So not all the minerals in the soil would be able to be absorbed, but plants have another method. And that is they use something called active transport. And this you should have done in your section on cells as well. Active transport occurs when special carrier molecules use energy to get needed substances into the plant, even if it's from a low to a high concentration. So let's have a look at this. Here we have our needed minerals. This is the cell membrane here, and that is our carrier protein. The minerals attach to the carrier protein. You can see them here. And the carrier protein uses energy to move those minerals to the inside of the cell, and they can then move into the cytoplasm and be used for whatever it's needed. Important is that energy is needed, and active absorption is always against a concentration gradient, meaning either from a high to a high concentration or from a low to a high concentration, which is exactly opposite to what happens in diffusion. Well, we've done an introduction to looking at how plants deal with water, and we've started looking at the root. But before we go any further, I think your brains need a break. Welcome back from your break. Let's get straight into having a look at how water moves through plants, starting with what happens in the root. So this is what we're looking at, what happens in the root. Let's have a closer look. Here we can see a root tip, and if we enlarge it and put it under a microscope, we can see this thing here, which is your root hair. And remember, the root hairs increase the surface area, so there's more space for water to actually go through the cell walls into the root. Now, how does this movement occur? By osmosis. And you should have learnt about osmosis, which is when water moves from a high concentration to a low concentration through a selectively permeable membrane. But what have we discovered about osmosis? Same as with diffusion, it will sometimes stop when the concentrations are equal. So there is a certain amount of active absorption as well. So what we've looked at is how water moves into the root through osmosis. Once water has moved into the root hair, it then has to travel through the root to the xylem, which will transport it upwards. So this section here is the cortex with parenchyma cells, and you can see that the water is moving through the cells towards the xylem. The water can also move along the cell walls. The cell walls are a bit like blotting paper, and water can move through the cell walls fairly easily. This movement is also done by diffusion if it's moving along the cell walls, and osmosis if it's moving from cell to cell. It's got to get into the xylem here in the center. That's the xylem star. So the water is actually trying to get into the xylem star so it can be transported upwards. A problem is it's got to go through the layer called the endodermis. And the endodermis is an interesting layer because it gives the plant control over substances moving into and out of the xylem. So let's have a look. This is your endodermis, and the endodermis has a thick strip of impermeable substances in the cell walls. So if water's coming along the cell walls, it can't go through the cell wall Casparian strip because it's impermeable. It's got to go through the cell. And that gives the plant some sort of control. Okay, so that's important. It gives the plant some control over the movement of substances into and out of the vascular cylinder. It also gives another aspect. This is a cross section through a root, a photograph taken under a microscope, but it's only portion of the root, and you should be able to identify this is the xylem, that is the phloem, and this is the endodermis. That is the thickened wall of the Casparian strip. But you'll notice there's no thickened wall here. So what it does is it's easier for the water to move through this part of the endodermis, and it's actually channeling the water into the xylem vessels because that's where they're actually being transported upwards. The previous picture was looking at a cross-section of a root. This picture is looking at a longitudinal or a lengthways section of the root, so we can see what happens once the water gets into the xylem. So let's have a look. This is where the water comes in from the root hair. It goes through the cells, it goes through the Casparian strip and the pericycle, and ends up in the xylem vessels. And the xylem vessels, it will then be sucked upwards, and it will start on its journey to the leaf. If we have a look, this diagram summarizes. We've already looked at water going into the root hair, passing through the cortex into the xylem. And the xylem vessels then go all the way up the stem of the plant and take the water to the leaves. So let's have a look at what's happening there. Diagrammatically, this also shows water moving into the root hair, through the cortex, into the xylem, up the xylem, through the stem. Okay, that's that part there. And when it gets to the leaves, still in the long tubes of the xylem vessels, some of the water goes into the cells for photosynthesis, and some of it goes into the air spaces and is then passed out in transpiration. Now, what we're looking at now is a little experiment to show that this actually does happen. I'm not just telling you stories. And in this experiment, celery, 3 pieces of celery, are placed in different dye substances. So let's have a look. First, celery, green dye. Second, red dye. Third one, blue dye. These pieces of celery were put in these a day or so ago, and you can actually see how the dye has been sucked up the stem and has gone into the leaves, giving the leaves their particular color. It's not as noticeable on the green one, but it's very definitely noticeable on the blue one. Now, if we take that celery stem and we cut it across, this is more or less what we see. That's the outline of the celery stem, and these blue dots, those are exactly where the vascular bundles are containing xylem. And that the xylem vessels are blue, which tells us it's the xylem vessels through which the water is transported. So this proves that xylem transports water. Now, there's a bit of a problem because gravity pulls everything downwards. Have you ever seen a waterfall falling upwards? No. So how come water can move up a plant? And that's something that puzzled scientists for many years, and they think they've found, how does this actually work? So the water has to go in through the root, up the stem, and then into the leaf. So there are 3 theories that explain this movement: root pressure, capillary action, and transpiration pull, and we're going to have a look at each one of these. First one, root pressure. And what this basically says is it says that as water is sucked into the root, and remember it's sucked in by concentration differences, and the water goes into the xylem, what happens to the water already in the xylem? And the theory is that the water already in the xylem is then pushed upwards. And that is what we call root pressure. And if you look at this experiment, that's the level of water at the start of the experiment. And after 5 hours, you can see that the water level has been pushed up as water is being sucked into the root, and the water already in the tube is then being pushed up. Okay, this is root pressure. But it can't push water all the way to the top of tall trees. So the scientists had to find another explanation to explain how water got to the tops of tall trees. So next theory they looked at was something called capillarity or capillary action. Capillarity or capillary action. And what that says is that water moves up narrow tubes. And the narrower the tube, the higher the height to which the water will rise. And in this experiment, there are 3 tubes, wide one, slightly narrower one, and very narrow one. And they are simply placed in water, and water will rise up the tubes simply because they are narrow. And as you can see, the narrower the tube, the higher the height to which the water rises. And because xylem tubes are microscopic, in other words, they're extremely narrow, so water will rise to a reasonably high height in these narrow xylem vessels. And what we have here in this picture, there's your xylem vessels, okay, with, remember, there's space there where the two xylem vessels join, and the water is moving through the xylem vessels. And there are two things that cause the water to move up the xylem vessel. First of all is that's a water molecule, okay? That's the xylem wall, and water molecules are attracted to the sides of tubes by an attractive force called adhesion. So the water molecules will want to be next to the side of the tube, but water molecules also stick to each other. So if one water molecule moves, it'll pull the other water molecules up behind it. And those are forces of cohesion. And that explains why water moves up narrow tubes. But that still didn't explain it properly. So the third thing scientists looked at was maybe when plants lose water through their leaves by transpiration, maybe that kind of sucks water up the stem. So they found out it actually does. And that's the third force that we're looking at, which we call transpiration pull. And that's basically as water moves up the stem into the leaves lose water, and that creates a suction force pulling water up the stem. And you can think of that as that's exactly what happens when you drink cool drink through a straw. You suck water out of the top of the straw, and the water already in the straw moves upwards. So this diagram illustrates water moving upwards, and remember it's been pushed up by root pressure, and then it goes into the leaves, and as it moves out in transpiration, the water already in the leaves is sucked up. And they've actually proved by experiments that this does happen. And here's one experiment that is actually used to show how strong this transpiration pull or the suction force of transpiration can actually be. So what we have here is we have a plant with lots of leaves, so it can lose lots of water. It's set up in a watertight system. No air can get in, so that as the twig loses water, the water is sucked up the tube, and the mercury is then sucked up the tube as well. And you know that mercury is pretty heavy, so to be able to suck up mercury, that suction force of transpiration is actually quite strong. What we're looking at now is the whole picture. How does water move through the plant? Right from in the roots, the water moves into the xylem vessels by root pressure, and it's pushed up by root pressure. Then, as it moves up the narrow tubes by capillarity. Then when it moves into the leaf and the water moves out, that's transpiration pull, sucking the water up. Then, not only is water moved through the plant, Once food is made, food also has to be transported, and that is transported in phloem. But transport in phloem goes in the opposite direction to water. So let's have a look. The leaf makes the food. The leaf makes the food. The food goes into the phloem. It moves down into the roots, and it goes into the root cells where it is stored. Before your brains get ready to burst, maybe we need an ad break. Welcome back from your break. I hope your brains are now bright and ready for the next section. Now, in plants, questions are often asked in regard to different experiments that have been done on how plants function. And I know that section is quite difficult for some of you, so we're going to do a little bit of explaining on the scientific method. And in your scientific method, there are two very important aspects. You first need to know what some things called variables are. Then we will have a look at how scientists report what they've known and the best way to write these terms, which I'm sure you've all heard of, but you might find are not particularly easy to do. Okay, so let's go straight on and have a look at scientific skills. You can sometimes be given pictures of experiments you've never seen before, and you have to be able to work out what is going on in them. And to be able to do that, you need to have an understanding of how scientists work. Okay, and that is called scientific skills, and that's what we're going to just have a look at to try and help you understand. Scientists work on something called the scientific method. They look at things. They observe. They wonder, I wonder why that's that color, or I wonder how this works. But they do more than wonder. They actually investigate, and they do experiments to collect evidence so that they can say, aha, I know how this works. And there's a certain pattern or method that they use to be able to do this. First thing is, when you do any experiment, you have to know that there are things called variables, things that can change or vary. Okay, and they are factors of an experiment which can cause change. And there's three types of variables that are important, and one is the independent variable, and that is the one that you change. You decide you're going to change this to see what happens. And what will happen because you've changed that is called the dependent variable, and that's the one that you measure at the end of an experiment. The third type is fixed or controlled variables, and these are variables that could mess up your experiment. So you've got to keep them constant if you want your experiment to be a fair test. And I'm sure you've heard of a fair test before. Let's give an example. There are these 5 boys in grade 10, and they don't really believe their teacher knows what she's doing. And she's told them that the brighter the light they put on their pot plant, the more water the pot plant will lose in transpiration. And they decide, let's check and see if she is telling the truth. So they decide to do an experiment. And for this experiment, they first have to look at what are they going to change, what are they going to measure, and what are they going to keep constant in the experiment. And so what they decided they would do is they would change the amount of light. Put the pot plant there, give it a bright light, or put it in a dark room and see how much water it loses. So what they would measure is how much water the plant is losing. Then, to make sure that the experiment actually works, they need to look at what other factors could affect transpiration, and those they need to keep the same. Okay, and from our lesson on transpiration, we know that temperature affects transpiration, so they would have to keep the temperature the same. Humidity affects transpiration, so they would have to keep humidity the same. Wind affects transpiration, so they would have to have the same amount of air movement. And then lastly, they must use the same plant. Because if they used a different plant, it might have a different number of leaves, which means it would lose more or less water. So they would have to use the same plant for that. Now, if there is a change, as they make the light brighter, the plant does lose more water, they will know our experiment is working. The change in what you measure, which is how much water is lost, is because they have changed the amount of light. But that only works if the other variables are kept constant. For example, if they put it in a hot room and a cold room, it might be losing less water because of the temperature, not necessarily because of the light. Then, what scientists do is once they've done the experiment and they're like, yes, it works, they have to write a report so that they can share their findings with other people in the scientific community. So they write a scientific report. And this I'm sure you are familiar with, because in your natural science, whenever you've done an experiment, you've actually come in contact with these different terms, and you should know how most of this works. So, they have to follow the same format, and the first thing the scientists say, what is their aim? What are they actually trying to do? Now, how do scientists write a scientific aim? Okay, there's a set pattern for how they write. For example, there is an investigation word. They then refer to the independent variable and relationship word, how it's going to affect what they are measuring. So let's look at that in terms of the experiment that the boys did. The investigation word was to see if. It could be to determine whether, to find out if. Then they mention their independent variable, which is the brightness of the light. Then they mention the relationship word. In other words, what's that going to do? Will it increase or decrease? Then, what are they going to measure? They are going to measure transpiration. Then, another one which I think everybody hates, and that is something called a hypothesis. It's a very big word, but it is actually quite simple. Because when you're doing an experiment, a hypothesis is what you think will happen, what you predict will happen in your experiment. And you know what? If you're wrong, it doesn't matter. In fact, sometimes scientists learn more from when their experiments don't work than when their experiments do work. Okay, so we're looking at a hypothesis here, and it's a smart guess. Doesn't have to be smart. It can just be a guess, but it's you or the scientist thinking about what the result of the experiment will be. And it's always written as a statement. So, for example, our boys, if they wrote their hypothesis, they would say, right, increasing the brightness of the light. That is your independent variable. Will increase. That's your relationship word. And what are you going to measure? The amount of water lost in transpiration. So if you look at this, it's actually very similar to the aim. It's just expressed slightly differently. Then, the rest of the scientific method you should be quite familiar with, because when you've done experiments, you've looked at the method, the results, where you write down, does it change color, or you might measure something. If you measure and you get figures, you would use a table. Okay, so you can have results. So there's method, how you're going to do it, results, which can be observations or measurements. And measurements are often recorded in a table, which makes it easier to read, or even a graph, which means you can look at the graph and you can understand how your experiment is working. Then, next one, conclusion. Okay, and in the conclusion, you put down your thoughts. What has actually happened in this experiment? Has it worked? Has it not worked? Is my hypothesis correct? What can I do to actually, if I do it again, actually make it work? So let's have a look at our boys' conclusion. Okay, so the boys' conclusion could be the brighter the light, the greater the transpiration, or it could be expressed in a different way as an increase in light intensity, which is the same as saying the brighter the light, and the rate of transpiration, which is the same as saying the amount of water lost. Both of these, though expressed differently, would be perfect for conclusions because they're mentioning the independent variable, the dependent variable, and the relationship word. So, what we've looked at are variables and the scientific method. After the break, we're going to look at an example and work out how to answer questions on experiments like this. Welcome back from your break. What we are going to do in this part of the lesson is we're actually going to look at how you can use what you have just learned about scientific method, variables and things like that to actually be able to answer specific questions that might be asked of you. I'm also going to try and help you to understand how to, if you are given an experiment that you've never seen before, how can you look at it and work out what is happening so that you can actually answer the question? Remember, any question you are asked has to be related to the content matter that you have done. So, what we're going to look at now is we are going to be using our knowledge of variables and how to write scientific reports, more specifically, what is an aim, what is a hypothesis, and how are they different, and then how to write a conclusion. And from what we did in the first half of the lesson, you should be able to see that they actually are linked quite closely together. So, what we are doing now is we're looking at an experiment that was done. I'm going to explain it in quite a bit of detail and help you to actually follow through so that you can think out the answers to the questions. So, let's have a look at the experiment. Okay, here's a picture of what has been set up. 4 fresh leaves, equal size, from the same plant, were treated in different ways. One had no petroleum jelly. Now, petroleum jelly is actually Vaseline. Okay, Vaseline is a type of petroleum jelly. So please don't get confused by petroleum jelly. It's simply Vaseline. So leaf number 1, no Vaseline on it at all. Leaf number 2, upper surface covered with Vaseline. Leaf number 3, lower surface covered with Vaseline, and the last one, both surfaces covered. Now, you might think, what will putting petroleum jelly or Vaseline on a leaf, how will that affect anything? The important thing to remember is petroleum jelly will not let air or water pass through it. So basically, in this leaf, we are sealing the leaf. We are blocking the stomata so nothing can get in and out of the plant. In this one, we are blocking only the stomata on the lower surface. Here we are blocking the stomata on the upper surface, and here there's no blockage of the stomata at all. Now, let's see what happens after a few hours. And you straight away going to think, oh, this is easy, but there's a catch, because the leaves are not in the same order as in the picture. They have been rearranged, and you basically have to work out which of these leaves here, A, B, C, or D, which of those has no petroleum jelly, both surfaces covered. So this test to see if you can understand what is actually happening in the experiment. So let's have a look at the types of questions that could be asked. First of all, in this experiment, what are your variables? What is your independent variable? And remember, that's the variable that you are changing. What is the dependent variable? And that is what you are measuring. And what are the fixed variables? And in this case, you should be able to find at least 2 fixed variables. So let's have a look. Independent variable, what are you changing? Which part of the leaf is covered? Dependent, what are you measuring? You're looking at the leaf to see how much it has shriveled up. Has it kept its shape or has it shriveled up? Fixed variables. What do you need to keep exactly the same? And here you need to think of the other factors that would affect transpiration. So you keep the leaves in exactly the same conditions. For example, they must all have the same temperature. They must all have the same light. They must all have the same humidity. They must all have the same wind. So that you know that it's not the temperature, light, humidity, or wind that is actually causing the leaves to behave in a different way. Then the last one is you need to have leaves that are basically the same size to start, and you need to have leaves that are from the same plant. And we'll talk about later why you need to have leaves from the same plant. Then, next thing we're looking at is, well, what is this experiment trying to prove? What is the aim of the experiment? And just before the break, we talked about how to write an aim. Then, the second question is, well, which leaf is the one that was covered in petroleum jelly on both sides? In other words, you have to look at the experiment and think about how it is related to what you have just learnt in transpiration. And give a reason for your answer. So, let's look at the aim. So, in the aim, you're looking to see. That is your first word, your investigation word. Okay, then your second one is which surface of the leaf, top of the leaf, bottom of the leaf, loses the most water. So that's your relationship, loses the most water. And the independent variable is transpiration. Then, looking at number 3, which leaf had petroleum jelly on both sides? And if you look at the pictures, it would be the leaf that has not lost any water. So it would obviously be leaf A. And the reason for your answer is leaf A has not lost any water. Then, more questions are asked. For example, which leaf had no petroleum jelly? And that should by now be quite easy to work out. It's got to be leaf D, because that poor leaf has shriveled up completely. In other words, it's lost more water than any of the others. So it obviously was the one that had no petroleum jelly on it. Then the next one is give a valid conclusion. Okay, so if we look at this, we can see D. Why is it D? Because it lost the most water. So it must be the one that didn't have any Vaseline on it. Then give a valid conclusion. Okay, and here you have to think out. What is actually happening here? What are we actually trying to prove? What is this experiment telling us? We can see that leaves that have Vaseline on it lose less water. But if you have a look at these two leaves, they both had Vaseline on one side of the leaf. One was the upper side of the leaf only, the other one was the lower side of the leaf. And they look different now. The one has shriveled more than the other. Which is the one that has shriveled the most? The one that had, oh dear, Vaseline on the upper surface or the lower surface. And the one that shriveled most is the one that had Vaseline on its upper surface only. Because most plants have more stomata on their lower surface than on the upper surface. So if the lower surface is open, it's going to lose a lot of water. This one, the lower surface was covered in Vaseline. The upper surface wasn't. So it lost some water through the upper surface, but not as much as leaf C. So your conclusion is all about which side of the leaf loses most water. And here you could say leaves with the lower surface covered by petroleum jelly lost the least amount of water and wilted or shriveled less. And that tells us that leaves lose most water through their lower surface, which means that there are more stomata on the lower surface. So this entire experiment is all about looking at which surface of a leaf loses more water. It looks as though we've run out of time, but in the lesson notes there is one more question for you to answer on the scientific method and this particular experiment. See you next time.